And the road to discovery continues. We know it's not possible for all of you in this room to make it out to Long Island to the lab. So we thought we'd bring a little bit of the magic of the lab here to this ballroom. And with me on stage are two of the young scientists who represent a host of unbelievable research that's happening at the lab. And I want to introduce, first of all, Mikola Engelblad, who is an associate professor and cancer researcher who's focusing on the microenvironment of the cancer tumor. Specifically, Dr. Engelblad's research, Engelblad, forgive me, I keep adding an N in there. Engelblad's research is looking into the role that cells play in fostering the growth of cancer cells. She's got some really cool stuff that she's going to show us because she's really at the forefront of advanced microscopy and has some rock star videos that we'll see in just a moment. What's really impressive is her work is being recognized as truly making a difference and being cutting edge. And earlier this year, she was named the recipient of the Era of Hope Scholar Award. That's from the Defense Department's Breast Cancer Research Program. So we're delighted to have you, Mikola, on stage with us. Thank you. Also here on stage with me is Michael Ronimus. Dr. Ronimus is a research assistant professor at the lab. He focuses on the genetic component of the autism spectrum disorders. He has just finished a multi-year study of the genomes of more than 2,500 families who are suffering and dealing with the challenge of autism. It is work that is being supported by the Simons Foundation. Among the findings so far, and literally just this week, there was a wonderful publication with your name on it in the Journal of Nature, the discovery of more than 100 gene mutations that cause autism. That is a truly important step that can help lead to much earlier interventions for children with autism. So, Dr. Ronimus, thank you as well for being with us. Before we get into the details of your research, just give us a sense of how you ended up in science in the first place. Mikola, how did you decide to do this?
Both my parents are physicians, and it was very natural for me to go to medical school. And in the second year, we had a course in molecular biology. And we had a book from Cold Spring Harbor Laboratory written by one of the people that's in the audience today, Dr. Tom Maniatis. And that course really was opening the world to me for a whole new way of looking at how diseases could be understood. And I quit med school and started working in laboratory on my PhD thesis on what molecular pathways are involved in killing cancer cells, breast cancer cells.
So it was literally a book written by one of the doctors in this audience?
No, it was the course where we used that book.
The course that you used that book.
It wasn't just a book, but the book was very important to us. We used it a lot. We called it either the Bible or Maniatis.
Wow, that's
Maniatis.
The Bible. Okay, Mike, what about you? How'd you get into this?
Well, it's a bit of a different story. My parents weren't physicians, but I grew up in the middle of the woods on a dirt road, literally, which is, I guess, rare in this day and age. And I spent a lot of time reading when I was a kid, and I became very interested in science. And I knew from a fairly young age, probably about the time I was 8 or 10, that eventually I was going to become a scientist, and that's what my interests were, and that is what happened.
One of the ironies, Mikola, is your work turned out to be very personal. As I said, you're really focusing on the environment in which the cancer cell can survive, but it turned out to be something that you had a personal imperative. I have to know why. Tell us a little bit about that.
Yes, so what you're seeing here on the screen is my family tree, and most of the pictures are from a very happy event, my confirmation. It's me in the bottom, in the middle, 14 years old with glasses. And what I didn't know...
It got better, didn't it?
What I didn't know then was, and you'll see that in the next slide here, is that my family is really hit hard by cancer. And so it became the work I do not just about the curiosity and finding out how things work, but really trying to see whenever we find something, is there a way you can turn that into something therapeutic? And it became that there came certain urgency to it, not just for me, but for so many other families that look like mine.
Wow, wow, that's an unbelievable number of family members that have been, and not uncommon. I'm sure there are many people in the audience who have a similar family tree. Mike, if we were having this conversation 10 years ago, talking about your work, we'd be talking about plants and some of the stuff that Bruce was talking about just a moment ago.
It's worth mentioning at this point, Cold Spring Harbor has a very good plant program, which I spent some time in. But in 2005, my oldest child was diagnosed with an autism spectrum disorder, and at that point, it changed my life, and I decided to say, whatever talent I have, I'm going to spend and expend is probably a better way to put it, in trying to contribute to whatever I can in understanding this disorder or set of disorders, which we don't understand at that time at all, really, and for which we have better understanding. I was very fortunate to be in a place and to be aware of a research project with Mike Wigler that was funded by the Simons Foundation, to be able to make that move and then to be able to contribute to that in a meaningful way.
So we see your little boy up here on the screen. He is absolutely cute as a button. Delicious, I would say. So you're at the lab and you're working on the plant side of things. This happens, and Cold Spring Harbor is the kind of place where Mike Wigler, I can go to his lab and ask him the questions. So you're asking as a concerned father, first of all,
right,
and then you dive into the research side.
Right,
How hard was that to do? How easy do they make it to switch gears?
Sometimes it's very easy. There were some hurdles which had to be overcome. But I mean, genetics is genetics. The genetics of plants and the genetics of humans, amazingly enough, is not incredibly different. I mean, humans have a brain, plants don't. That's one big difference. But the genetic principles that underlie both are quite similar. So it wasn't difficult to understand the principles involved in moving to human genetics at all.
So tell us a little bit about your work. What are you doing? We've got a slide that just came up with, looks like some coding that you've done.
Well, there have been a lot of technological advances that have occurred in the era of genomics since the human genome was sequenced and since microarrays and next-generation sequencing became commonplace. So one of the first studies we did, or the first major study I was involved in, was looking at very large-scale mutations in the human genome. So we do this by looking at families, and this is from the Simon Simplex collection, which was put together in very comprehensive fashion. It is a tremendous resource, even now. And we looked for mutations that are present in children, but not in their parents. And by looking for these new mutations, we're able to identify hotspots in the genome. And what this shows are the human chromosomes, and the colored dots on top of them and below them are hotspots that we found that contain large-scale mutations. And in some cases, when you see multiple dots there, or ovals, I guess they are, those are things or regions in which we see multiple mutations. So these are things that definitely cause autism when they're found in a human.
And you've identified at least 100 different gene mutations that are responsible for autism.
There are different ways to interpret that number. I would say we've conclusively identified dozens, and we've identified a lot of candidates, hundreds of candidates. And additional work will narrow down that list until we estimate there are about 400 to 500 genes that, when disrupted, can cause autism. And in the next few years, we hope to be able to narrow that list and conclusively say these are the genes that cause it.
And Michaela, tell us about your work and what you're learning about the environment in which cancer is able to flourish.
Well, so as I said, my parents are both physicians, and one was bringing along a lot of X-rays, and the other one a lot of ultrasounds. So I really was, I guess, conditioned to looking at disease. Disease was something you could understand if you were looking at it. So what we built was this technique where we literally superglue a mouse to a glass slide and put it on a microscope, and then what you can see from that is...
Can we put the thing on the big screen too, please, up here in the middle?
These
videos?
two movies over here, to this side, would be a movie where you can see an untreated tumor. You can see the cancer cells in blue, the inflammatory cells, immune cells in green, and then blood vessels in yellow. And there's really a lot of activity there just in a tumor. This is 300 times sped up from normal real time. Mm-hmm. But then what you have on the other side, we started to treat a tumor, and you can see we're using a trick. So all the dead cancer cells switch color from blue to red, a little bit like, I guess, the electoral map last week. And you can see the inflammatory cells are coming in huge numbers, all these green cells. And what we found is that the cancer cells almost using, is sending out signals to turn these green cells, that's part of our immune system, from, so to speak, Anakin Skywalker into Darth Vader. They make them go so that they start secreting survival signals from the cancer cells and make it harder to treat them.
So you're trying to figure out how to turn off the switch that's allowing these cells to do this.
So this actually allows us two strategies to try to get the therapy we already have and already use to work better. One is to prevent that they come in the first place. We found some of the switches for that. And the other one is to try to prevent that the cancer cells put them over to the dark side of the force.
How close are you to finding that? What is the support that the people here have been able to bring, doing to facilitate your being able to get to that next hurdle? I mean, this is unbelievably impressive right here.
So we have identified some signals. Some of these are now in clinical trials. We are trying in mouse models now to find out which of these strategies we think will work the best. Are there better ways to use it? Will it work in all types of cancer or only breast cancer? So there's still a lot of hurdles to overcome, but I think we're excited about it.
One of the things that I realized when we were having our conversation earlier was your work is informing one another. What you're doing in cancer is informing what you're doing in the genomics of autism and vice versa. Can you give us a little sense, Michael, of how that works?
Well, sure. I mean, the techniques that we use were largely developed initially to look at cancer. And by refining those, we look for the types of mutations we find in autism, which are much harder to find. We're looking at genomes that are more normal, if you want to call it that. In a tumor, it's very messed up. So we refine these tools. We find the mutations that are really hard to find, and then this is sort of synergistic. This comes back to cancer again. Then we need to refine them out of necessity. They come back and can be applied to cancer and give even greater precision in looking at the types of genetic events that go on in cancers.
With what you know right now, can you help your son? Can you help other children with autism, what you've learned so far?
We're laying the groundwork to be able to do that. So I think down the road, in the relatively near future, an autism diagnosis won't be just the opinions of a number of psychologists or developmental pediatricians. It will be more, you give them a genetic test and we say, this child is at risk. These are the therapies that might work for this particular child. And if you can intervene during a very early developmental window when the brain is very plastic, you can produce a really optimal outcome. And there's a lot of evidence already that suggests that's the case. So we can get in there sooner and we can do the right things as opposed to sort of doing a hit or miss approach.
And Mikaela, what about your work? Are you able to see a point where you can stop those inflammatory cells from feeding the cancer tumor and it metastasizing?
There's a lot of excitement in the field right now because by targeting other types of immune cells here the last couple of years, we're starting to see cures in patients with advanced metastatic cancer. It's just a small number so far, but it looks really promising. And so we're all invigorated by this whole new way of looking at tumors, not just the cancer cells, but looking at them in the context of the immune cells and the inflammatory cells.
So ladies and gentlemen, this is the work you're supporting. Groundbreaking research in cancer, hopeful research in autism. Mikaela Egeblad and Michael Honmas, thank you so much. You've been wonderful. We're going to take a break. Enjoy your dinner. We'll be back with some awards shortly. Thank you so much.